Hi, I'm Jeff Hoy. I'm the Sugarcane pathologist with the LSU AgCenter. This video is intended to be part of a sugarcane field day. We'll have a field tour stop addressing two subjects, diseases and billet planting. And I'm going to talk about the diseases. Herman Wagaspak, the Director of Research with the American Sugarcane League, will talk about the latest information on billet planting. So with diseases, we try to focus on research that's going on the station, and that is right now occurring in two main areas. First of all, there is some research related to billet planting where we're looking at fungicides and an insecticide, dimethoxam, or the product called platinum, and can it reduce the severity of red rot, a stalk rot, that interact with stress conditions that occur after planting of billets to reduce stand establishment and ultimately yields. So we've had success. We have some labels now for fungicides, Quiltexcel or generic versions, Preaxer and Rebitec. So these chemicals applied alone, the fungicides can be helpful, and the insecticide, but they tend to be somewhat erratic depending on the conditions that occur after planting. The combination of the two is consistently helpful, but unfortunately the label's been submitted for the insecticide, but it hasn't come through. It may take even a couple more years. So in the meantime, we have these fungicides labeled, and anybody that's going to plant billet should go ahead and apply fungicide. Do so with multiple nozzle array on your planter and high volume of water, 35 gallons per acre, which is troublesome but essential to get complete thorough coverage. So that's the aspect related to billet planting. The other thing we do is the breeding program to develop new high-yielding varieties is probably the most important thing going on in the station, and I plug into that. We try to develop disease-resistant varieties. Resistance is a big component of higher yields. And we do this a couple of ways. Some diseases we do an inoculated test in the field, smut, leaf gall. We do this to try to identify the susceptibles and resistance, separate them out. We look for natural infection levels to distinguish the resistant from the susceptible in mosaic and brown rust. So this works. It takes a lot of effort and time, but it's also troublesome. The environment can interfere, and getting good results takes a while. So we've got another big project that's been ongoing. I'm working with Dr. Naranjan Bisak, who's also going to be on the program for the first time, talking about the increasing element of molecular breeding, importance of it in the breeding program. So what we're doing there is trying to develop what are called molecular markers. These are DNA segments that are associated with genes that are accounting for resistance in the varieties. And it allows us, in a way, if you can get them, you can track resistance in your population, say with your parents. Optimize the crosses, pick the best parents to get the highest frequency of resistance and the best levels of resistance in your selection population. If you have enough resources, even screen the selections. So we're making some progress in this area. We've got some markers for leaf gall that are already being put to work, applied in the program. Mosaic is looking promising. Smut and brown rust are coming along as well. So we hope we're going to see more of that success with that in the future. Last thing I want to talk about is sometimes we can't come up with resistance. And what's the alternative there? Well, sugarcane is vegetatively propagated. So if you plant stalks from an infected plant, you're going to spread and increase the disease along with the planting process. So we have to put some effort into giving healthy planting material for the growers. A healthy seed cane program. We've had a long-standing successful partnership with public and private sector effort now that's producing seed cane through a lab process called tissue culture. And we have a seed cane certification program, which is LDAF regulates, or making sure certain standards are met for quality. And this has been a very successful program. Has it provided better control of our diseases? Well, the simple answer is yes, but it's allowed us to grow some varieties that are susceptible to certain diseases, and that is a huge advantage. So the hardest challenge for the breeding program is to come up with varieties that can produce good stubble yields or tune yields on heavy clay soils. And the prime example is LO1 299, and that variety is susceptible to smut. We would not be able to grow it without a regular infusion of healthy seed cane. Tissue culture seed cane has provided that. We've controlled other diseases as well. We've grown susceptible varieties. So this program has been a huge positive impact on industry, and any grower that's growing sugarcane should be that should be a regular yearly component to your program, is to obtain and plant healthy seed cane. So that's what I've got for you today. See you next time.